Welcome back. We have a top concerning story today of the biodiversity effects on human health. Climate change due to human activities has been a cause for biodiversity loss, which is a major contributor to declining human health. We have co-evolved with microorganisms which live in networks within our bodies, which is modeled by the hygiene hypothesis. Society changes from a developing to a developed society. The environment is experiencing many changes.
The hygiene hypothesis, as stated by Hansky 2012 and Rook 2011, is the reduction of human exposure to natural bacteria causing adverse effects. The radical changes such as smaller family sizes, exposure to viruses, urbanized living, and increased antibiotics are changing the direction of the immune response. With this information, we conducted an experiment to test the influence of humans on microbial functional diversity across surfaces from low to high human involvement to determine the difference in more natural habitats and human-simulated habitats on microbiota. To test our hypothesis, we used ecoplates containing 31 different types of carbon sources commonly found in the natural environment that are used by microbes. This figure shows a map of an ecoplate outlining the distribution of the carbon sources throughout the 96-well plate. The 31 carbon sources and water control are repeated 3 times across the 4 columns so that the 3 samples can be run for each ecoplate. We collected 12 samples of possible microbes in the environment ranging from low to high human influence from a sample of soil to the bathroom counter and inoculated the plates. A tetrazolium dye turned purple when the carbon sources were metabolized. This helped us to determine the functional diversity by looking at the functional richness and evenness. Richness is measured by the amount of the 31 carbon sources that were metabolized, and the evenness is the intensity of metabolism in each carbon source. We used a sterile-cultured tube and phosphate-buffered water to collect a sample the size of a standard piece of paper. We pipetted the sample into the ecoplates containing the 31 carbon sources.
Plates in the picture is similar to our results. Richness is measured by the number of wells turned purple and evenness with the shade of purple. There is more purple with the lowest human influence locations. The higher human influence locations possibly utilize different sources of carbon, or the microbes cannot utilize the sources available. It is possible that the function of the microbes in different human influenced environments differs compared to the microbes used in a more natural habitat. Figure 1 shows the diversity index for low, medium, and high levels of human influence. Locations with the lowest human influence had significantly greater functional diversity than the medium and high human influenced areas.
And now we go live to Graham Rook, the author of Hygiene Hypothesis and Autoimmune Diseases from 2011, to hear about his work.
Thanks for having me, guys. So throughout the 20th century, there's been a shocking rise in chronic inflammation disorders. Changes in microbial contact has changed our immunoregulatory mechanism. This hypothesis was first stated by Charles Blackley in the 1870s when he noticed that aristocrats and artisans were more likely to develop hay fever than the farmers were. The thoughts of when humans became in less contact with these microbials started back when we went through a transition and became in less contact with feces, farm animals, mud, and pets. Before this, we had a lifestyle that had a major fecal-oral transition, but in return we became adapted to a number of animal viruses such as influenza B and C, smallpox, mumps, and the measles. Thanks, guys.
Thanks for that great information. We'll now go to Ilka Hansky to look at a specific study.
In order to test the hygiene hypothesis, I traveled to Finland and tested a random sample of school children between the ages of 14 and 18, all living in a 100 by 150 kilometer area. I collected 116 blood samples and screened them in order to look for allergen-specific antibodies. When we ran our tests, however, we found out that our results do not reveal the mechanism of environmental influence on atopy. We found out that only 5% abundance of the staphylococcus on the skin of individuals with atopic eczema versus only 4.4% in healthy individuals, which is not a statistically different result. Although our study did not find statistically different results, many studies in the past have seen that high density in microbiome is associated with low risks for allergen disorders.
And we're back with the results on the hygiene hypothesis. The results of our experiments are important to the general public because it shows that humans are in very little contact with much biodiversity. There was less species richness in high human influence places such as the UST building wall and the door handle in the science building. This low response means we humans are not in contact with microbes from other species-rich environments. The hygiene hypothesis talks about the recent change in our environment for our society, so much so that there is imbalance to our immune system because it's not challenged. The new sterile environment we are currently living in does not challenge our immune system by being exposed to microbes, and it doesn't help our immune system defend against infectious diseases that cause unwanted immune diseases such as asthma and inflammatory diseases that are chronic. Large recent study done by Hatella found that the annual prevalence rate of most disease clusters was lower in environments where people were living with more green space, 10% or more than average, within a 1 kilometer radius. The relevance for humans to be more exposed to a higher biodiversity correlates to human health and the ability for a better immune response. As effects from immune diseases increase, we as a public need to be informed about the importance of the hygiene hypothesis and how microbes are necessary in our everyday lives. Information about the subject can benefit the human population because knowledge about microbes and environment biodiversity is crucial in understanding more about diseases and to better protect our ecosystem.